Hello and welcome to the Deep Dive.
Great to be here.
Today we're diving into a really interesting paper. It tackles a massive challenge in structural molecular biology.
Yeah, how to actually see these incredibly huge molecular structures and simulations people are generating now.
Exactly. We've got some sources here focusing on a new piece of software designed specifically for this. So our mission is to figure out, what is this thing? Why was it needed? And crucially, what makes it different?
Let's get into it.
Okay, so maybe start with the problem itself. Why is visualization so important and what changed recently?
Well, visualization isn't just for making nice images. It's fundamental for understanding. How do molecules fit together? How do they move? You need to see it.
Right.
The issue is the data size has just exploded. We're talking cryo-EM giving atomic detail, better AI prediction tools, more accessible simulations.
So we're dealing with systems way bigger than before.
Oh, yeah. Millions of atoms are now routine. And some models, like these coarse-grained whole cell simulations, are pushing towards 100 million particles or even more.
Wow. And existing tools, they just can't cope.
Pretty much. They're often fantastic for smaller systems, don't get me wrong, but load up one of these massive trajectories and they slow down, they freeze, sometimes they just crash. They weren't built for this scale.
Yeah, I think anyone in the field has experienced that frustration. You load something big and just watch the wheels spin.
Exactly. So there's a clear need, a gap, for software designed for this molecular big data.
Okay, so that sets the stage for VTX.
Precisely. VTX is presented as this open source, high performance tool built, from the ground up specifically for real-time visualization of these massive systems and their dynamics.
High performance and real time, those are the key words. So how does it actually achieve that? What's the magic under the hood?
Well, the core is its graphics engine. It's built on modern OpenGL, and it's heavily optimized for rendering huge amounts of molecular data very, very quickly.
Okay, and they mentioned specific techniques. Something about meshless representations. What does that mean practically?
Right. So think about drawing an atom, like a sphere or a bond, as a cylinder. Traditionally, software builds a 3D mesh, like a net of polygons, to represent that shape.
Okay.
That works okay for thousands, maybe tens of thousands of atoms. But scale that up to millions or 100 million. Building and managing all those tiny meshes becomes a huge bottleneck. It just takes too much memory, too much processing power.
So meshless avoids building all those polygons.
Kind of, for things like sticks, ball and stick,
Really?
and spheres. VTX uses cleverer implicit methods. It tells the graphics card how to draw the shape directly at the pixel level without needing that complex pre-built mesh.
Ah, so it's much more direct, much more efficient.
Exactly. Massively reduces the load, especially for these enormous systems. It still supports other representations too, like the popular cartoon view for proteins and SES, which is the solvent-excluded surface.
Got it. More efficient rendering. But does it still look good?
Oh yeah. They've included standard high-quality rendering options, good shading, ambient occlusion for depth, fog, outlines, anti-aliasing. Plus, you can export really high-res images, up to 8K, performance and quality.
Nice. What about actually using it, the interface, the controls?
Yeah, they seem to have put thought into that too. There's the standard trackball camera rotation most people are used to,
Right.
but they also added this really interesting free fly mode.
Free fly.
Yeah, like in a video game. You use keyboard keys, like WASD to fly around inside the structure. Apparently, it's super useful for navigating these huge, complex systems.
I can imagine. Especially with like a whole cell model.
Definitely. And they added keyboard shortcuts for common things, helpful if you're on a laptop trackpad. The whole interface is modular, too. You can dock panels wherever you want.
And file formats. Does it read the common stuff?
Yep. Seems pretty comprehensive. PDB, MOL2, MMCIF for structures, and trajectory formats like DCD, XTD, ARC. It uses the ChemFiles library in the background, which is quite robust, plus direct PDB download.
Okay, that's convenient. But the real proof is how it performs against the established players.
Absolutely.
Hmm.
And this is where the paper gets really compelling. They did direct comparisons.
Who did they test against?
The big ones, BMD, PyMol, and CameraX. They use benchmark systems ranging from 100,000 up to that 100 million particle whole cell model.
And the results?
Quite dramatic, actually. On the 100 million particle system, PyMol and ChimeraX essentially crashed or froze immediately. Couldn't handle it.
Okay. What about VMD? It's known for handling large-ish systems.
VMD could load the 100 million system and also a big 11 million atom system they tested, but crucially, once loaded, it basically froze. You couldn't manipulate it, couldn't rotate the view smoothly in real time.
So loaded but unusable for actual interactive work.
Exactly. Whereas VTX, even running on what they describe as a consumer laptop,
Yeah.
could load these massive systems and allow smooth real-time interaction, rotating, zooming, selecting things, changing styles. They even showed video of using that free-fly mode inside the 100 million particle cell model.
That is a night and day difference. Being able to genuinely interact with a system that size in real time, that's a game changer.
It really seems like it could be. It potentially opens up analysis for systems that were just too big to explore interactively before.
So who gets to use this? Is it
Mhm.
available?
Yes, it's open source, free for non-commercial use. They provide builds for Windows and Ubuntu, and the source code is up on GitHub.
Great. So researchers, students, educators can just download it and try it out.
Seems so. It also has some standard analysis tools built in, like measuring distances and angles, doing structural alignments.
And are they planning more features?
They mention ongoing work,
Yep.
things like enhancing the real-time rendering further and adding Python scripting support, which would be huge for automation and custom analysis.
Okay, so wrapping this up, what's the big takeaway here?
Well, VTX looks like a really powerful, necessary new tool. It directly tackles this major bottleneck in structural biology, visualizing truly massive data sets.
Its performance on systems where other tools struggle seems to be the key differentiator. That optimized engine, the meshless rendering.
Right. Combined with the usability features like the free fly mode and its open source nature, it feels like a significant step.
It definitely raises a question then, for you listening. If we can now truly interact with, fly through, and analyze these previously unmanageable molecular cities in real time, how might that accelerate discovery? How could it change our fundamental understanding of complex biological processes?
Something to think about.